It's going off of. Oh.
Yeah.
Okay. Now I need to do. Hold on. I'm going to do run this first.
I can also turn off your camera as well. If you don't.
Okay.
What's your name? Hey, Jordan. Okay. We met at the
Yeah. Correct. And so let's try to share.
Here we go.
Yes.
And let's talk to
Yeah.
Okay.
Amazing.
Right.
Yeah.
Yep. So now we're receiving this
Great.
presentation.
Perfect.
The only thing that's going to
Antonio.
That's good. Sometimes the floating meeting controls that pop up on Zoom, sometimes they show up
Oh yeah, yeah.
there.
But they're not now.
It blocks everything, but looks like we
Okay.
got that. We've got
Great.
situated.
Is this the room mic?
That is the room mic.
Okay.
Oh,
I could just speak up or tell people to come up to the front, and there's a few people here.
so the gooseneck is this one.
Okay.
So that should now be
We're good.
good.
Okay.
All right.
Is there an introduction? Do I just start? Okay.
It's not. Here's myself. Great. Set to go. Right on track.
Yeah.
Thank you.
Where's the office space?
Just in the health sciences center, like that way, a half mile, I guess, kind of in the by the Plaza Cafe. All right, so it's first floor beyond the T-wing, the northwest wing is better. It's so in the basements, yeah, kind of close to the main hospital entrance and down two floors.
So we're doing some renovations there.
Yeah, doing some
Something that would turn down people think.
It looks like, and it's interesting, like you go downstairs and you can see it, and it's like, oh, it looks like a really nice little plaza, like residential area. It's all like fenced off still. I have no idea what they're like, and it has been like since summer. So, I don't know what they're still working on.
All right, let's get started then. Welcome everyone to today's seminar. So today we're very pleased to have Dr. Brian Schertz, who is Associate Professor of Laboratory Medicine and Pathology at the University of Washington. And he's also, as of last year, an affiliate professor in our department. So Dr. Schertz's research focuses on diagnostic testing and communication between physicians, genetic counselors, patients, and families about the results of these tests. His group seeks ways to improve genetic outreach by using modern resources to help relatives connect and communicate with each other. Brian, the floor is all yours.
Thank you. Can everyone hear me in the back? There's plenty of space if you want to come closer. It seems like I walked in and heard that there was two competing meetings going on now. So I appreciate everyone who's here and everyone online. I think we have all of like 4 people online, maybe more. Well, maybe more than 4 people, maybe even 6 people online. But I appreciate you coming, listening to this talk on a Thursday afternoon. I hope it's useful and interesting to you. I'm mostly going to talk about hereditary cancer. Like the title says, I'm going to talk about identifying clinical ascertainment bias, which is what some of the focus of some of my statistical work has been doing. The other piece, which I don't think is interesting in terms of a statistics view, is classifying variants of uncertain significance, which is pretty simple Bayesian math when you come down to it. I think this has some more interesting statistics, so I wanted to present it. And I've been working on these projects for several years and have never presented them. So I'm really glad to be able to present in a forum like this. Wearing my clinical hat, people look at this and are like, oh, that's really important, but kind of we don't really get it or are not really interested. So let's see if I can advance slides. We're gonna see.
Pick one with the mouse first.
Pick ones with the mouse first. Okay. Oh, okay. So just as a overview, I didn't put a conflict size. A conflict of interest, I do some consulting for a group called Constantium Biostatistics. It's not something that I'm going to talk about here. And I do clinical testing as a job. And then the other disclosure, I guess I should say is that I'm not going to be at the University of Washington as of December. I'm actually moving to Vanderbilt. So if you want to talk to me about this later, come soon. And so I can give you my email at that institution. Okay, so we're going to talk about two things. One is bias in genetic cancer assessment when we talk about family ascertainment. And then we're going to talk, so first we're going to talk about bias when it comes to ascertaining families. We're talking about genetic risk. And then we're going to talk about bias when it comes to ascertaining clinical cases when it comes to looking for genetic risk. And of course, families are really key to looking at genetic risk. I'm going to talk, here's a picture of Mary Claire King, who some of you may know, in 1994, right when she was identifying the BRCA1 gene, localizing it to chromosome 17. And that was really a groundbreaking story because it showed for the first time, it proved for the first time that cancer wasn't just totally random, but that there are heritable components to cancer. Now, since then, we've found probably, well, over 20 really solid genes, and then some other genes that have cancer risk kind of as a secondary thing. But still, only about 5 to 10% of cancer is heritable. When I say heritable, I'm talking about a cancer risk where one gene causes like a 3 to 50-fold increase in cancer risk for a specific type of cancer. And then we can talk about familial cancer, which is where you get polygenic risk scores, which I know are genetic. But there's also familial factors that are not genetic, and it's hard to tease those two apart sometimes. And so maybe a third, between a quarter and a third of cancer is familial or hereditary. And then most cancer is sporadic. Most cancer really is chance, environmental risk factors, random mutations. And for the rest of this talk, we're going to talk about this preventable hereditary cancer. And I say hereditary cancer is important because most of it is preventable, partly because individuals who have hereditary cancer risk, like hereditary breast cancer risk from BRCA1 or BRCA2, hereditary colon cancer risk from Lynch syndrome, they can take, there's separate guidelines because the cancer risk is so high. And so people do really dramatic things like have complete mastectomies, which decreases the risk of breast cancer from about 50% if someone has BRCA1 or BRCA2 pathogenic variant to closer to 5 to 10% risk, which is actually a little bit lower than the risk in the general population for women. And there's other cancers like endometrial cancer. If somebody has Lynch syndrome, if a woman has Lynch syndrome, they have about a 40 to 50% risk of lifetime risk of endometrial cancer. And if that woman, after she's done having kids, has her uterus taken out, then the risk goes to zero, like absolutely zero. No one who doesn't have a uterus gets endometrial cancer. It just doesn't happen. And so there's prevention, and increased colonoscopies or other increased screening can really dramatically reduce the risk of cancer. Okay, so now we're going to, any questions about that introduction? So now we're going to talk about a gene you might not have heard as much about that contributes to hereditary cancer risk, and that's CDH1. Has anybody here raised your hand if you've heard of CDH1 as a cancerous gene? Okay, so CDH1 is a gene that's typically been known as a gene for hereditary diffuse gastric cancer. And this is a brief overview of the studies that established this. So initially, this FAIR study in 2001, they followed the same path. They followed the same path that Mary Claire King had done to find the hereditary breast and ovarian cancer genes, where they said, okay, we want families that are enriched for cancer. And we're going to take those families and we're going to, because those are the families who are more likely to identify the genes that, if there are genes that are associated with this risk. And gastric cancer is an interesting thing because it's been thought of for a while that there's very big differences between populations in gastric cancer risk. And so it's been known for a long time that there are environmental factors that contribute to this. People eating food that has a lot of nitrates and a lot of salts looks like it might increase the gastric cancer risk. But it's been hard to pin down the exact risks that are associated to different environmental factors. It's really hard to figure out diet and pin down exactly, exactly what dietary things people have been eating for many years. And so they took a bunch of families and identified this gene called CDH1 that was very clearly associated with gastric cancer in about a third of the families where they found these clusters, a lot of gastric cancer. And so these are the risks that they're looking at. So the Farrow 2001 article, then they took those families where they identified CDH1 as the gastric cancer. And I'm not going to get into linkage analysis. Take a class in linkage analysis. You get a family and you get the pedigree and then trace which chromosomal segments have traced through what to the individuals who have the disease, and then which ones are most likely to have this. So they narrowed it down to CDH1, found a bunch of truncating mutations with gene CDH1. It fit the story. It's involving kind of gastric endothelial lining growth, and then so they found these initial estimates, and from those initial studies, they said that people who had gastric cancer, people who had CDH1 pathogenic variants, had between 56 and 83% for women, and between 40 and 70% risk of gastric cancer by age 75 to 80, depending on which numbers you're looking at, which study you're looking at. That's a really high risk of gastric cancer. And that's a high enough risk of gastric cancer that based on these numbers, the consortium got together and said, we're going to have a recommendation that in a family that has hereditary diffuse gastric cancer, and in individuals that have pathogenic CDH1 variants, we're going to recommend a complete gastrectomy. So that's taking your stomach out. Not having a stomach is compatible with life, but it has some pretty severe life changes. You have to be on some multivitamins, and your stomach's not just involved in holding food, but also involved in releasing hormones that help digest the food and help absorb nutrients. Even though most nutrients are absorbed in the small intestine, so people who have gastrectomies have in addition to being a pretty dramatic surgery, but with some immediate risks, have a long time kind of lower survival. So what they found was that in these families that had pathogenic CDH1 variants that had hereditary diffuse gastric cancer, then most people said, okay, the surgeon says I should get a gastrectomy, I'm going to take my stomach. And some people said, I'm going to take my chances. I'm going to take my chances and see if I might gastric cancer, because living a life with a stomach and having a risk of gastric cancer seems better than knowing that I'm going to live a life without a stomach. So in addition, there were some questions about how the study was done, and so then later on, there's a follow-up study, and they had slightly different guidelines for this study, where they were looking at they just had two people who had confirmed hereditary confirmed diffuse gastric cancer, and then one person didn't have to be certain that they had hereditary diffuse gastric cancer. So they got these groups to classify as hereditary diffuse gastric cancer, found the one people who tested CDH1 positive, and got these results, which are a little bit lower, 40% risk for men, 63% risk for women with point estimates. This is a very small study, only about 7 families, so the confidence intervals are enormous. Then in 2015, 8 years later, they did a hamster study, changed the guidelines a little bit, but still required at least 2 individuals with gastric cancer in the family. And here they found 70% risk for men, 56% risk for women of diffuse gastric cancer. So that's the story. And there may be students in here, right? So I want to pause here and have you think, are these risks right? We've got multiple studies, peer-reviewed studies over 15 years, and we'll come back to these. Okay. In the meantime, 2013. Here's another picture of Mary Claire Higgins. Yes.
Can you know something about survival?
Yes. What about survival? That's a great question about survival. Yes. So diffuse gastric cancer is an awful cancer to have because it's really hard to detect. There's no really good screening. In order to screen for it, you have to do multiple different gastric biopsies. So you have to stick upper endoscopy, stick a tube down someone's throat, take gastric biopsies, take those, have the pathologist look at them. And if somebody actually does have like hereditary diffuse gastric cancer, you get about a 50% chance of finding it. Because it's diffuse, meaning it spreads out a lot, and it's let me go back to here. It's diffuse, meaning it's spread out a lot, and it can be patchy. So like it's not like you detect one, you get one part of the stomach and you got the right one. You have to like get lucky and get the right part of the stomach that it goes to. So there's no really good screening for diffuse gastric cancer. It's not like mammograms where you can just take a, mammograms are painful. I've heard, haven't had one. And are not fun, but they're pretty good at seeing where the thing is. Or colonoscopies. Colonoscopies are fantastic because not only do you go up and see what's there, and I have had a colonoscopy, also not fun, but you go and see like where the precancerous polyps are, and you take those polyps out. So it's not just a good screening test for cancer; it's also prophylactic treatment for precancerous lesions. There's all sorts of other, but gastric cancer, like ovarian cancer, is one of those things that there's really no good screening for it. And the other thing about diffuse gastric cancer is diffuse gastric cancer spreads really, really fast. And so people who start having symptoms of gastric cancer are oftentimes dead within 2 or 3 years. So you can see why someone would want to get their stomach out, because it's like, if I have an 80% chance or up to 80% chance, or even 50% chance that I'm going to get a cancer that's going to kill me in 2 years, I'm going to do some pretty extreme things to be able to prevent that. Did that answer your question?
Yeah.
Yeah. Okay. So getting back to the story. So feel free to raise your hand and pause at any time for questions. We've got plenty of time. So this is a group of individuals who developed this broker cancer risk panel. Colin Pritchard was one who led this. Mary Claire King and Tom Walsh in Mary Claire King's lab were integrally involved in this. There's Angie Jacobson, who was a genetic counselor, who's involved, and Steve Salaponte did the bioinformatics behind this. This was back in 2011, 2012. And they developed this panel. And actually, this wasn't the panel in 2012. In 2012, BRCA1 and BRCA2 weren't on the panel, but then the Supreme Court invalidated the BRCA1 and BRCA2 gene patents, and then we could put them on the panel. So the next day we said, oh, they're on the panel. So we're detecting all of these genes, and it's mostly designed for breast and colon cancer. We put CDH1 on the panel. And one of the reasons why I put CDH1 on the panel, as you may have noticed, is that there's increased risk for breast cancer in individuals who have pathogenic CDH1 variants as well, particularly lobular breast cancer. But only about 10% of breast cancer is lobular. But in these individuals, it seems like it's closer to 50 or 60 or 70% of breast cancer tends to be this. In lobular versus ductal breast cancers, a pathology, you'd have to ask a pathologist to see kind of what's the difference between those two. Both equally not good cancers to have. So we started doing this testing, and most of the people who are being referred to this testing are being referred for breast cancer screening, because breast cancer is a really common cancer. Gastric cancer, very, very uncommon. If I asked everyone in this room to raise their hands if they know someone who has breast cancer, I bet almost everyone raised their hands. Now, if I asked everyone in this room to raise their hand if they knew someone who had gastric cancer, I bet maybe one, if I'm lucky, person would raise their hand. So it's just common cancer versus uncommon cancer. From a statistics, I know this is a biostatistics group, right? It's not a very satisfying statistic. But I'm trying to illustrate gastric cancer never gone. Most of the people getting this test are getting tested for breast cancer. But what we found was that we started to see families that were like this. Clearly pathogenic CDH1 variant, and this is just an example family, but we saw it. So, and this individual is getting tested because they have breast cancer at 45. This person has breast cancer in their 60s. This person has breast cancer in their 60s. And when we test individually, see we have these pathogenic CDH1 variants, but no one in this pedigree has gastric cancer. And we asked to make sure everyone's asking about it, but this person has a 50% chance of having the same variant. This person has a 50% chance. These people have 50% chance of having the same variant. So there's probably, if in this family, at least 2 or 3 other people who have this pathogenic CDH1 variant. And so we know we have probably have 4 or 5 people who have this pathogenic CDH1 variant in this family, no gastric cancer. And we saw this a few times. And at a American Society of Human Genetics conference, I saw a poster of somebody presenting about some unique pathogenic CDH1 variants who worked at GeneDx, which is a big genetic testing laboratory, does way more genetic testing than we do at the University of Washington. Said, are you seeing the same thing? And they were like, yeah, we're seeing the same thing. We're seeing all these families, they're getting tested for breast cancer, and we might see one person with gastric cancer in the family or zero people with gastric cancer in the family. Like, does this seem weird to you? And they're like, yeah, this seems really weird to me because we thought that the risk for gastric cancer is like 60 to 70, like, the quote is up to 80%, but that's really not a good estimate because the point estimate was here was 80%, but the other numbers, but still 40 to 60% lifetime risk of gastric cancer in a family like this, we expect to see something, right? So now I'm going to pause here and I'm going to say, why aren't we seeing more people with gastric cancer in those families? And I'm going to pause and ask, especially the students, what are potential hypotheses? But what are your potential hypotheses for why we're not seeing more people with gastric cancer in those families? So shout out something in the audience. Yes. Yes. These are the only two people who have been tested. So we don't know about the other ones. Yeah. Now I would put negative if they didn't have it. So that's a great question. So they don't know about, and so we don't know about, so I'm going to see if I can do the pointer. We don't know about this person, we don't know about this person, don't know about this person's for this person. The people who are married in are very unlikely to have it. So between those 5 people, each of them have a 50% chance of having that variant. And so I expect, on odds 2 or 3, maybe they don't, but this is an example pedigree. We've seen like between that other laboratory and our laboratory, we knew of like just off the top of our heads, like 30 families that look just like this. And yes, so this is unusual to see this pedigree. And, between at least 3 and potentially up to 8 people on average, we're guessing probably 4, 5, 6 people who have the CH1 variant, no gastric cancer. So why are we seeing this? When the estimates are like this in 3 peer-reviewed published papers, why are we seeing families that don't have this? There's multiple hypotheses that are very good, legitimate hypotheses. So let's shout some out. You're all, are there PhD students here in biostatistics? Okay. I expect you to have at least some ideas. I don't expect you to be brave, but I expect you to have some ideas. So someone needs to speak up and give some ideas of why they think they might. Yes, go for it.
Based on what's up here, it looks like these studies use some kind of like outcome-dependent sampling, right? So they're sampling people who already have gastric cancer and then looking back at their like the genetic risk markers.
Yeah.
That's maybe we did more of a like prospective study. We measure people's risk markers and then track their future cancer process.
Yeah.
You can see sort of a different story. It sounds like that's kind of what happened when you were testing patients with breast cancer.
Yeah, that's a great hypothesis. And that's the hypothesis we're going to test in the next slides. So wonderful. So you got but I want to pause here though, because so we decided we wanted to test that hypothesis. We wanted to say, is this ascertainment bias, which is what you're describing, right? The short way of saying it, ascertainment bias. We're ascertaining people who have gastric cancer, at least in this criteria. This is the criteria established in 2015 for this one. And these ones, they had to have 3 people that had gastric cancer. And so for all these studies, at least 2 or 3 people in the family had to have gastric cancer for them to qualify for the study, for them to be classified as hereditary diffuse gastric cancer. And then they're getting those same families that have at least 2 or 3 people who have gastric cancer and saying, well, let's see what the lifetime risk of gastric cancer has in these families. So like the gentleman said, we're ascertaining people based on a phenotype and then saying, what's the risk of that phenotype? This is a classic example of that. This could be a classic example of ascertainment bias. But when we initially proposed this and talked to some of these people who had been kind of in these groups, they're like, well, maybe it's just there's different types of variants in the different families. Like maybe the variants in these families you're looking at are less penetrant than the variants we're seeing in these families. Or maybe there is other family things that are going on. And so like in some families, you have a bunch of environmental risk factors as well. And that plays with that interacts. There's a gene environment interaction there. with the pathogenic variants. And so in families that have this pathogenic variant and have the environmental risk, there's a lot of risk. And maybe the families you're looking at just don't have a lot of these environmental factors. And so they still said, well, if somebody has 3 relatives that have hereditary diffuse gastric cancer and a pathogenic CDH1 variant, then they still have this level of risk. Okay, so there's multiple competing hypotheses, because the people who did these studies are like, no, we want to take people's stomachs out, or it's important. We don't want people to die of diffuse gastric cancer when their risk is really high. Okay, so we decided to do a retrospective review of personal and family history, and in this we had 75 families that we found in GeneDx in the University of Washington that had pathogenic or likely pathogenic variant in CDH1. We used the standard guidelines to classify pathogenic variants. And this is what we look like. So we have 75 families, 52 unique pathogenic variants. This is what the variants look like. The most important thing about this is we said, if there's anything where anybody might have any qualms about it being a really clearly, fully pathogenic, totally kill the CDH1 mutation, the gene effect, we're going to exclude it. When people say, well, maybe they're different variants, we want to just say, no, they're the same variants. And actually, we looked at the other studies and said, we want the same types of variants. And a lot of these were the same variants that were found to be pathogenic, and these other studies that found high penetrance. Okay, so we wanted to address that. And then here's the demographics, just so you can see 1,650 relatives in the pedigrees. So we wanted people who had the ordering test on an individual, but clinicians will give a pedigree when they do this testing. And they give that pedigree so we know what they're testing for. We know what types of cancers in the family, and the molecular pathologist is providing the report, can provide a custom report that specifically addresses the risk in the individual and the risk in the family. And that's going to come up important later. So mostly large multi-gene panels, some custom panels, some specific CDH1 testing. Usually that's going to happen if there's a high risk, a high likelihood of wanting to get CDH1, or if there's a CDH1 variant already known in the family. So like the sister or the aunt, uncle, cousin has this pathogenic CDH1 variant. They're getting tested because they know about the specific one in the family. And then the management panel is also similar. So mostly large panels with some targeted testing. You can see the ancestry. This is pretty typical for genetic testing in the United States, unfortunately. There's pretty much everyone except white women is underrepresented in hereditary cancer testing. Here you can see the cancer status. So this is among the probands and relatives. So here's everyone. Here's the probands. Most people are unaffected. These are people that have any cancer. So it could be breast cancer, it could be gastric cancer, it could be colon cancer, some of these other cancers as well. But most people are unaffected, which is consistent with most people getting hereditary cancer screening as well. This is all family members. So there's about 50% of the families, there's no cancer reported in the family at all. And so you can see where the results are going already. And here you see kind of how much there's cancer in the families. So we're not seeing the cancer we expect to see in these families just by chance. And likewise in the probands, people are getting tested because they have it, but there's still a lot of probands. Sorry, there's still a lot of these 20% of probands are getting tested without having a personal history of cancer. And this is the proportion we see. So of the cancer that we see is 22% gastric cancer, 78% no cancer. You can see the diffuse gastric cancer versus not otherwise specified. Diffuse gastric cancer is just a subset of gastric cancer that's specifically associated with CDH1. Sometimes it's not, that data is not available. In the women, 151 women, a lot of people had breast cancer, some not. You can see the portion of lobular breast cancer to ductal breast cancer. That's like more than flipped. Like I said, usually it's about 10% of breast cancer is lobular breast cancer. So to see this much lobular breast cancer is really interesting and consistent with CDH1. Okay, and then how many of them met criteria? So some people met criteria, but a lot of people didn't meet this criteria for their families. Okay, so then we used this. This is John Renault. I don't know if any of you know John Renault. He was here about 6 years ago. He was a postdoc in biostatistics. I hired him because he was great and he was interested in genetics. And so we only had about 41 families where we knew there was complete pedigree diagrams. We didn't want pedigree diagrams that were like only had the people who had CDH1 pathogenic variants. We wanted, or only had cancer. So sometimes when genetic counselors draw pedigrees, they only draw the people who are exciting and don't draw the people who are boring. We wanted to make sure that we had complete pedigrees that had exciting people and boring people in and I'm not saying we interviewed them to see if they were boring or not. You know that, right? Just not an interesting clinical history. So what we did is we counted the individual's gastric cancer in each pedigree, we excluded the proband to reduce ascertainment bias. We wanted to say, the proband we know is ascertained because there's something about them why they're getting genetic testing. So we're just going to look at family members. And then we're going to compare the actual number of gastric cancers to what we expect in those families. We're going to use a maximum likelihood estimate. And this is just a little more details about the statistics because it's about a statistics lecture. So we're looking at individuals and we expect that every year they're going to have a certain risk of cancer. So we just add up the cancer. This is like the same you do for adding up cumulative risks. So we're saying what's the cumulative risk of cancer in this person based on the SEER statistics. And then for a group of individuals, we do the same thing across a group of individuals. And then for a group of individuals in a pedigree, this is the interesting thing. So in a pedigree, there's an individual who we know has the variant, but we don't know who else has the variant in the pedigree. Well, sometimes we know one or two other people have it. Like if we know that their sister has the variant, then we can give their parents a 50% chance. If we know our cousin has the variant, then we know that one of the parents is an obligate carrier and the other parent's an obligate carrier, and one of the two common grandparents has it as well. And so knowing who has the variant, who doesn't have the variant in the pedigree, we can infer what the chances are that the other people in the pedigree have the variant. For an individual, it's an awful guess. Like I could say, you have a 25% chance of having the variant and you don't have cancer. What are the odds that you don't have cancer by chance? What are the odds you don't have cancer because you having the variant? So from an individual standpoint, it's not a very good way of kind of making any predictions about someone for their clinical management. But from a statistics standpoint, it's great. You can say, given that this person has a 25% variant, which doesn't exist in nature, what are the odds that they have cancer? If they have the variant and it's pathogenic, and for any given risk for that specific variant. So that's what we did for these pedigrees. We took these pedigrees and we said, here's what the pedigrees look like, given the number of people who have cancer, given the number of people in these pedigrees and the ages of all the people in these pedigrees, how many gastric cancers do we expect to see? And based on the population statistics for gastric cancer, and what increased risk would we need to have to see this number of pathogenic variants in the pedigree. And we use maximum likelihood to be able to do that. And these are the hazard ratio curves we generated. And I'm going to show you three hazard ratio curves here. One is we took all of our data here. And then for this one, we took only families where there were at least 3 people with gastric cancer in the family. And the reason why we did this is because we knew what other people had done, and we wanted to say, do we replicate their findings? We know we're purposefully ascertaining based on 3 gastric cancers in the family, and we're going to see if that bias gives us the same results as we saw in the other studies. And then we took the rest of the people who didn't have 3 gastric cancers in the family. So here's the risk curves, so we see about, I can't read the numbers there. I have the number on the other slide. About 40% risk for male and about 25% risk for female. I think it's the point estimates. And this is a huge risk of gastric cancer. This is 40 times the population risk of gastric cancer. But it's a lot less than the 91 times risk of gastric cancer that we see in the people who have at least 3 cancers, and the families have at least 3 cancers. Just using those pedigrees, and about closer to the 27 times risk of gastric cancer. So here's the numbers looking in a different way. So here's our numbers. Our gastric cancer in the whole population is 36% with these confidence intervals, and females 20% with these confidence intervals. And you can see that these are like significantly lower than the gastric cancer estimates that were seen with the ascertained families that are ascertained based on gastric cancer. And our numbers for over 3 are actually right in line with what those numbers are, which gave us encouragement that like what we're doing is actually mirroring what they were doing in the lab. And if we mirror what they're doing, the other studies have done, then we're gonna be fine. So the coolest thing about all the studies, I think, is that the breast cancer risk is remarkably consistent across all the studies. Why is the breast cancer risk so similar across all studies when the gastric cancer risk is so different? And I think the answer is really simple. Nobody ascertained on breast cancer. We didn't ascertain breast cancer. They didn't ascertain breast cancer. And so there's not the same bias for breast cancer risk. So the conclusions of this is cumulative risk of gastric cancer was estimated to be 36%. 36%. This is lifetime risk at age 80 for men, 20% risk at age 80 for women. This is significantly lower than previous. It illustrates why ascertaining on families can be challenging. Prophylactic gastrectomy. It's going to be a more difficult conversation now. Unfortunately, it becomes a more difficult conversation now than it was before. If you know that the risk is like 60 or 70%, then a surgeon can come in and say, I highly recommend you get your stomach taken out. And somebody has to have a pretty strong will to be able to say, no, I don't want that to happen. If I come in and say, your risk is probably 20% lifetime risk of this awful cancer that will kill you, or I can guarantee that you have no stomach, then it's a difficult decision. So now we have a much more difficult decision to make, a much more difficult conversation. But I believe is a conversation that's more that's reflective of more accurate statistics. Okay. This is my midpoint question. I have a picture of the Sesame Street, Goldilocks and Three Bears. So this is one I want people to be brave. And I want to ask the audience, is our gastric cancer risk prediction, the one that we made, is it more likely to be too high, too low, or just right? There's only three options. We take a minute to think, and I want everyone to raise your hand, okay? You don't have to raise it very high. You can raise it like this if you want. I want everyone to raise your hand, just to think about this. Okay, so how many of you think the risk that we calculated is more likely to be too high? Raise your hand. How many think the risk is likely to be too low? Raise your hands. How many think the risk is likely to be just about right? No one gets just about right. That's okay. If you didn't raise your hand, come on. Okay, this is great. I think that most of you who raise your hand about it being too high are right, and I'll show in the next half why that's true. But before I show that, this study just came out last month, or 2 months, 2 months ago. Anyway, this came out in September. It's November now. They looked at CDH1 variants and lifetime risk of cancer in a much, much larger study of people that weren't quite ascertained clinically in the same way. There was still some clinical ascertainment, and the risks they found were even lower. So you need to have like glasses. I'm sorry, I just put this on. So this is about 10% risk of gastric cancer in men and about less than 10% risk of gastric cancer in women. And then the risk of breast cancer is actually still a little bit lower, but pretty close to what we were guessing before. And so the risk, we were guessing like 40%, but the risk might be even lower. So why might that be? It might be because, let me go back here. We're getting people who are, which one was it? Here we go. We're getting people who have known CDH1 pathogenic variants. So we're ascertaining on families that have already been tested for CDH1 for some reason, right? So this whole chunk of people who've already been testing for CDH1 for some reason. Some of these ones are also people who are having this custom panel who they're looking just for gastric cancer. These are the ones that are more like the ascertainment that we saw on that other study. So a good chunk of people we're testing for might actually have been getting testing because they had gastric cancer in their family and because there was a high index of suspicion for gastric cancer. There's another reason that it might even be too high beyond that, and I'll show you that for the next few slides. And this is something they also show, and I looked into the literature, looked into the paper a little bit to see if this is actually wrong or not, and I can't tell for sure. They said in these families, there's higher risk than in these ones, but I couldn't figure out how they could exclude these individuals while still calculating the risk, right? So I'm not sure if this curve is actually true or not, or if this is just like saying, look, there's low risk if there's no one with gastric cancer, and there's also low risk if there's no one with gastric cancer. And when you have people with gastric cancer, then all of a sudden you see some risk, which like, I'm not sure if this curve is the right curve. I'm not sure if this curve is not just obvious. It's like, look, you have a lot of people in the family and it looks like there's high risk. So I think I don't think that this graph actually is meaningful, but they may have done something clever that I didn't know about. So we haven't completely proven that there's not environmental factors that might influence this, is what I'm saying. There may be environmental factors that are family specific that make some families more likely to get gastric cancer, or it could just be chance. Some families happen to, if there is 10% risk of gastric cancer, some families by chance are going to have 3 people have gastric cancer. And some families by chance are going to have 0 people have gastric cancer. And the families that have 3 people with gastric cancer are of course going to look like they have higher risk than the families who don't have anyone with gastric cancer. And the only way to parse those things apart would be to have a really large prospective study and follow up over time. That's the only way to figure that out. And I'm not sure if there's a way and no one's taken the time yet to do that perspective. This study was retrospective as well. Yes.
Have you looked at CH1 variants preclinical models and see like, oh, when we do like a CRISPR knockout on CDH1, we see gastric cancer risk in mice goes up by X amount. Like, is there evidence there?
Yeah, there have been mouse studies, and I don't know about them very much. So I do know that like this risk here, this 10% risk of gastric. So we said that in the families, let me go back to here. We said in the families where there's only 1 to 2 people with gastric cancer, then we get 27.1, here we go, 27.1 times risk of cancer. This is a huge increase in risk. And sorry, 47.3 in the whole cohort. This risk here is 30%. They've said 30%, 30-fold, not 30%. This 10% risk is 30-fold lifetime risk of greater than the general population. And so, yes, like this is a super high risk of gastric. No one's saying that CDH1 is not a gastric cancer risk gene. Everyone agrees this is a gastric cancer risk gene. This causes an enormous increase in risk in gastric cancer. It's just not 80% or 60%. It might not even be 30%. It's lifetime risk of gastric cancer. So, okay, did that answer your question? So like we know that pathogenic CDH, no one's arguing that they're not pathogenic, and there's plenty of biological evidence to show that this gene is really involved in this pathway. Okay, so we're gonna talk about Lynch syndrome now. Does anybody know Lynch syndrome? Maybe you've heard of Lynch syndrome. Lynch syndrome is a little more common than CDH1 CDH1. So it used to be called hereditary non-polyposis colon cancer when I was in medical school. Then they said, hereditary, yes. Non-polyposis, well, what they're saying is it's not familial adenomatous polyposis where you get hundreds and thousands of polyps, because you only get a handful of polyps. So it's not polyposis, you only get a handful of polyps, but that was really confusing because you do get some polyps, those polyps go on to get cancer. So that didn't really work. And then colon cancer was found in colon cancer, but it also causes increased risk of endometrial cancer, ovarian cancer, and several as small bowel cancer, stomach cancer, several other cancers as well, brain cancer. And so like, well, they were moving away from like naming syndromes after people. So they call it a hereditary non-polyposis colon cancer. And they said, well, you know what? The name doesn't work. It's not descriptive enough. And we don't want to name it, 3 pages long describing everything. So it went back to Lynch syndrome. This is John Lynch, who first identified the genes involved in Lynch syndrome. 4 genes are involved in Lynch syndrome: MLH1, PMS2, MSH2, and MSH6. Let's see, this is MLH1 and PMS2 form dimers, and MSH2 and MSH6. Here, MSH3 also forms a dimer, but MSH6 causes higher cancer risk, also forms a dimer. And all 4 of these proteins are involved in mismatch repair. And so when the DNA replicates, there can be a mismatch, and the machinery goes back and corrects that mismatch. And so that's how they're involved. And these are real pedigrees from real people who have drawn to show kind of what types of cancerous you look like. So this is one where there's a positive colorectal cancer MLH1. Person gets colorectal cancer at 56. There's a bunch of colon cancer in the family. And you can see that you can also get renal cancer and brain cancer as well. Here's another family. This person has colorectal cancer at 31. And now they're 47 years old. This person had polyps diagnosed at age 40, but is still alive and hasn't developed colon cancer. Only about 50% of the people who have pathogenic variants in these genes get colon cancer. Actually, a little less. This is one in MSH6, so it's less than the penetrance is lower. I'll show you the penetrance later on. But there's quite a bit of colon cancer in these families. These are the type of pedigrees that I told you about, that only exciting people are drawing these pedigrees. Usually there are a lot more people in these families that are not shown. So this is what let me advance. in the canvas. Okay, this is what the risk looks like, and I've cut and pasted this together from 4 different pages from the National Comprehensive Cancer Network guidelines. And I've cut and pasted this together so you can see for colorectal cancer, MLH1 and MSH2 have, I said, about 50% lifetime risk of colorectal cancer. MSH6 and PMS2 have a little bit lower penetrance. Colorectal cancer, 4.1% in the general population. Endometrial cancer, 3.1% in women, 0% in men, or in people who have no uterus. I should say that's the more correct term. Endometrial cancer, up to 50% risk, so 30 to 50% risk for MLH1 and MS2, a little bit lower risk for MSH6 possibly, and certainly lower risk for PMS2. Ovarian cancer also much higher risk in the general population. Renal pelvis or ureter cancer. It happens more often than you'd expect by chance. We don't know exactly what the risk in the general population is. It's hard to tell exactly what fold risk there is here. Bladder cancer, we also see more often gastric cancer, small bowel cancer, pancreatic cancer, prostate cancer. And then brain cancer, we also see more common in these individuals, but it's very rare in the general population. And right now they say see footnote M for all of these. And what they say in footnote M is there's a lot of studies that suggest that there may be increased risk of breast cancer, but it's controversial. So that's what I'm going to tell you about now. Is there breast? So the question we're going to ask is there increased risk of breast cancer in people with Lynch syndrome? Now, some of these cancers, like biliary tract cancer, it's really hard to know the fold estimate of risk. It's really hard to prove that there's increased risk because we know we see them pretty frequently in individuals who have Lynch syndrome, but it's still a pretty low percentage. And we know that people with Lynch syndrome are getting screened more often and might be more aware of cancer. And for cancers that are very uncommon in the population, it's harder to figure out what the exact fold increased risk is because it's hard to get a really good point estimate of that risk in the general population. If there's any ascertainment bias in your study, then you might get a much, much higher risk than you see in the general population. And so we suspect there's higher risk of many of these cancers. Breast cancer is the opposite issue. In women, the general population risk of breast cancer is between 8 and 12%, depending on who you ask, which makes it like the most common cancer of women. This is a very, very common cancer. And the risk is so high that in any cohort, you're going to see a lot of people who have breast cancer who don't have whatever risk factor you're looking for. So in this cohort, it's the same thing. When we see breast cancer, is it because of ascertainment? Or is it because there's actually increased risk? We know that for Lynch syndrome, there's not 10-fold risk or 5-fold risk like we see in BRCA1, BRCA2. But is there increased risk is a question we're asking about. Okay, so this is one study that came out and said, what we think that there may be increased risk of breast cancer specifically for PMS2 and MSH6, not for MLH1 and MSH2. Let me go back. So they're saying these genes don't cause increased risk of breast cancer, but we think these genes might, is what this paper said. And what they did is they took a bunch of people with Lynch syndrome, they monitored over time, and they said, this is what we see. A bunch of people that not people they monitored, sorry, a bunch of people who tested positive for Lynch syndrome in clinical laboratories and said, well, let's look at those pedigrees to see how much breast cancer and ovarian cancer and other cancers. But let's see how much breast cancer is in these individuals who have Lynch syndrome. And we know they have Lynch syndrome because they're getting clinical testing, and we're seeing that they have Lynch syndrome. We're looking back at their histories to see what cancers they have when they're diagnosed with Lynch syndrome. And so A is breast cancer, B is colon cancer, C is ovarian cancer, D is endometrial cancer. Colon cancer and endometrial cancer are cancers that we know are associated with Lynch syndrome. Ovarian cancer, there's strong evidence that it is. Breast cancer, we're not sure about, and that's what we're looking at to see the questions. And so you can see MLH1 and MSH2 have a lot of colon cancer, as PMS2 and MSH6. Here, endometrial cancer, MLH1 and MSH2 stand out higher. We see more people who have pathogenic of these genes that are getting ovarian. But here, we're seeing kind of more ovarian cancer for this MSH6, relatively more breast cancer for these individuals with PMS2, and also with MSH6. So these are less represented. And so they use this data to say, well, because of this, we think that maybe MSH6 and PMS2 are associated with breast cancer. another study did something similar. They're clinically ascertained families. They're saying here's MLH1, and here's the proportion of women who have all of these cancers. And of course, we see lots of people with colon cancer in all of these groups. And we see lots of people with endometrial cancer in all of these groups, because those are the cancers that we know are associated with Lynch syndrome. But what was interesting is that in these groups, MSH6 and PMS2, there's a lot of people who have breast cancer. And 30%, 35% of people have breast cancer. That's way higher than the percent of people who have breast cancer in the population. So they estimated like a 2.6-fold increased risk in breast cancer, not in these individuals. This is not significantly different than the prevalence of breast cancer in the general population. This is about what we expect in the population. But in people with MSH6 and PMS2 mutations, there's a much higher risk of breast cancer, they said. And this is the family members. So this is not looking at the individuals, but looking at the families. And in the families, we see something similar. Not quite. This is the average number of cases per family. So this is not a risk in any way. This is a number of cases per family. And they say, look at in these families, we see more cases of breast cancer than we see in these families. So maybe this is in line with the likelihood there's increased risk for breast cancer. So the weird thing about this is that if you go back and look at these, I thought these were the bad genes to have. These are the ones that have higher risk for pretty much all the cancers on the list. And these ones are the ones that have lower risk. Why are they causing increased risk of breast cancer? Okay, I'm going to pause here and let you think. I'm not going to have you speak out or raise your hands even because we're almost out of time. But you can think kind of why are these people having breast cancer? So what we did is we said, maybe it's the ascertainment bias. Because that's the theme of the talk. So maybe it's ascertainment bias. So what is the ascertainment bias? And what we did is we went and said, okay, we're going to get a whole bunch of pedigrees, and we're going to assume somebody has a pathogenic variant. And then we're going to say, what's the probability that we have a bunch of breast cancer, a certain number of breast cancers in that family, a certain number of colon cancer. We're going to get SEER data, which is like the general population data for cancer risk. We're going to say, based on the age of the individual family, we're going to just randomly say, how many breast cancers do we expect? How many colon cancers do we expect? And so then what we did is we said, we're going to take these families and we're going to insert one or two, in this case, we're going to insert two people who have cancer. And we're going to say, of all the families that could be out there, we're only going to take families that have at least two people with cancer in them. If someone has less than two people with cancer in it, we're not going to take those families. We're just going to say, you know what? Clinically, if someone has no history of cancer, why are they coming to the hereditary cancer clinic? And why are they ordering genetic testing for cancer? And this is actually true. When somebody comes into the hereditary cancer clinic to see if they qualify for genetic testing, the genetic counselor is going to go through and say, how much cancer is in your family? If there's no cancer in your family, we're not going to order genetic testing for hereditary cancer. And if there is cancer in your family, we're going to order genetic hereditary testing for you. And so what we did is this is what we saw. So this is what looks like for MLH1, and this looks like so there's zero risk, 5-fold increased risk, 10-fold increased relative risk, 15-fold relative risk. And this is we looked at kind of the log p-values to say if we ascertain families that have at least two people with risk, when there's really no risk, but we only ascertain families where there are at least two people with cancer, what does the risk look like? And it looks like there's significantly more risk in these families. This should not be surprising, right? And no matter how much actual risk there is, if we only ascertain families that have at least two people with cancer, the risk looks higher. Does that make sense? Should make total sense. Yes, you're only getting people that have the trait you're looking for ascertained. And so we went back to our broker panels and we said, we have some data about this. What does the ascertainment bias actually look like? We happen to have a bunch of families that were ascertained based on either testing for colon cancer or colon testing for breast cancer. And we can go back to the clinical notes and say, this genetic counselor ordered this because they said they were testing for breast cancer, or they said they were testing for colon cancer, right? And so we just looked at cases that were said testing for breast cancer or cases. And so these are ordering codes where they're putting why they're ordering the test, because insurance wants to know why someone's ordering the test in order to pay for it. So genetic counselors are pretty good at saying, we're ordering this test because we have a family history of this, or we have a personal history of this. And this is what we saw. And this is the money slide. And I'm going to walk you through it in 5 minutes. So these are all families with negative genetic testing. And I know for a fact that we're pretty close to like 95 to 99% good at getting all the common causes of hereditary cancer. So we're pretty confident that everyone here does not have any hereditary cancer risk. There might be a chance we miss something, but over 90% chance we miss something. So this is testing ordered for breast and ovarian cancer risk. And you see the black line is the number of cancers we see in the families. This is about 20 families, I think, that we did this on. And then this is a simulation saying for all of the for the pedigree structure, how many cancers do we expect by chance based on their ages and based on the pedigree structure? Again, negative families. And this is how much expected. So we see way more breast cancer in families where they're ordering a genetic test to see if there's hereditary breast cancer. This should be obvious. This is telling us that what we're doing is right? They're ordering, they're being ascertained for breast cancer, they're testing for breast cancer, they have more breast cancer than you expect by random chance. Ovarian cancer, much more ovarian cancers than we expect by chance. Endometrial cancer, more than we expect by chance. Which is interesting because genetic counselors are taught about syndromes for endometrial cancer are different than syndromes for ovarian cancer and breast cancer, and they're not an overlap. Colorectal cancer, we see more colorectal cancer by chance in families that are getting testing for breast and ovarian cancer. Prostate cancer? Not at all. At this time, during this period of time, there was no indication that prostate cancer is hereditary at all. It's changed since then. But when these tests are being ordered, no indications. So prostate cancer is extraordinarily common. By far the most common cancer in men. Many men have prostate cancer and die of heart disease. Pancreatic cancer is also associated with BRCA1 and BRCA2, so we see more than that than expected by chance. But why is there more colorectal cancer and endometrial cancer in families that are ordered for hereditary? And so here we see the same thing in people getting testing for colon. These are Lynch syndrome families. Families that people think there's Lynch syndrome. We see way more endometrial cancer than you expect by chance. We see way more colorectal cancer. Well, more colorectal cancer than you expect by chance. And it's not quite significant, but we also see more breast cancer. We're seeing a trend. Our numbers weren't big enough to be able to see a significant difference in the amount of breast cancer. So what it seems like is happening is that some people come to the clinic and they have more cancer than expected by chance in their families. And if someone has a lot of cancer in their family, regardless of what kind of cancer it is, the genetic counselor is going to say, I'm going to order this test for cancer. And if there's more breast cancer in the family, they're going to say, well, I'm going to order the breast cancer one. If there's more colon cancer in the family, they're going to say it. But they know that our panel has all of it on it. And so they're going to say, you know what? This doesn't, and sometimes they'll say this family doesn't quite meet criteria for breast cancer, doesn't quite meet criteria for colon cancer, but it's close on both of them. We're going to order the testing. And so what we're seeing here is what I'm very confident we're seeing here is we're seeing ascertainment. These numbers actually mirror almost exactly the numbers we expect from our data, about 2.6-fold increase in breast and ovarian cancer in people where colorectal cancer testing is being ordered. So the bottom line is there is a subtle ascertainment bias in clinically ascertained pedigrees. Now, remember, for all of these, we excluded the proband. For every single study I showed in the last one, we excluded the proband. It's not the person getting testing, it's their family history that we're looking at. And people usually would think, oh, if we exclude the proband, we're getting rid of the bias. But in clinical ascertainments by genetic counselors and medical geneticists, they're getting family histories. And some family practice doctors are as well. So there's clinical ascertainment bias, not just in the probands, but in the families. And those prior genetic associations are most likely due to it. So why are we seeing an MLH1 and why are we not seeing it? Let me go back here. Why are we seeing it in, here we go. Why are we seeing it in MSH6 and PMS2 and not in MLH1 and MSH2? Because these families have a much clearer history of colon and endometrial cancer risk. These are families where the risk is low enough that you don't see as much colon and endometrial cancer in the families. Here's the colon cancer, right? And so it's more likely they're being ascertained on breast cancer or some other things that they're moving on to genetic testing. So I think that's the most parsimonious explanation. If you want to argue with me, that's great. I hope there's time for questions. I'm sorry I took up so much time. But one thing we know how to figure out if there's clinical ascertainment bias. What we don't know is how to resolve or adjust for that clinical ascertainment bias at the end. And that's all. Thanks for your time. Oh, I forgot my, hold on. I didn't do my thank you slide. A whole bunch of people were involved in this. Let me do my thank you slide. I have to do the thank you slide. Here we go. Here's a lot of people who are involved in this. Mostly John Renola, who you saw. He's the John McEnroe. He's working at Ambry now, a phenomenal statistician, and really did a lot of the heavy lifting for all of that. Lots of other people as well. So thanks. Questions? You have like a minute.
Yeah, I have a question. So
Yes.
you mentioned the JAMA paper. How do the type of results make it into clinical practice? I mean,
Yeah.
how does that change physician recommendations typically, and how long it takes to?
So for the CDH1 story, then it took about once our paper was published, another paper was also published at the same time. So this had similar findings, but didn't get actual risk estimates, but pointed out the issues. And so those two papers together changed clinical practice within about a year. It took about a year for people to read the papers, people to digest them. But it's still changing. So this was like 4 years ago that it was published, and there's still debate going on. And I think this last paper was published a month ago is finally going to put the nail in the coffin and say, no, we can't say, over 50% risk of gastric cancer. It's probably closer to 10% risk. So it's taken 2 papers to start changing it, and then another 4 years for someone to do a bigger paper to really confirm it. And that's actually very fast. Sometimes it takes 7 or 8 years for things to get clinical practice. How did the papers of ascertainment bias get into the journals for high impact journals in the first place? Well, the first paper got in because it found the gene. So it was a really important paper for finding the gene. They never should have done risk calculations. Somebody should have said, take that piece out. It's a biased number. And they put a disclaimer in there saying, this is the risk only in families who have at least 3 people with hereditary gastric cancer. So they were saying, like, but it's also just like who have the statistician who's reading the paper, did not did not do a good enough job of saying, this is not the right number. Okay, good question. Other questions? Yeah. What did? Oh, the gastrectomy. That started in, let me go back, 2001 was when that it was shortly after this first paper in 2001 that recommendation came out. By 2007, it was kind of the standard recommendation. And so for between about 2005 and 20, I can't remember when our paper came out. So for 15 years, that was the standard recommendation.
That's very fun. You continue monitoring them, like data on like what the eventually given not.
Yeah, I'm a pathologist and statistician, so I'm not a gastroenterologist, and I know that people are starting to do those studies now, and look at these families prospectively. It's as you think about it, it's an even more difficult decision for families now, because you tell somebody you have a 30-fold risk of gastric cancer, and that risk is 10%, which is a super high risk of gastric cancer. And that gastric cancer, if you get it, is like 90% likely to kill you, to lead to death in 2 years. And so it's the decision between it's really like you need. This is why genetic counselors exist, to help people walk through those decisions in a paternalistic way and help people kind of think through that. It's a really difficult decision. I'm glad I don't have a pathogenic CDH1 variant in my family because it's a challenging thing. Yeah. Yes.
Do you know how good MRI is for screening?
Not very good.
Not very good.
Yeah. They've done studies on MRIs to see how good it is, and it's not very good. This is a difficult one. Yeah. Yeah. Yeah, ovarian cancer is similar. It's really difficult to do screening for ovarian cancer. And so many times it's one of the most gastric cancer is more deadly, but ovarian cancer is also a very deadly type of cancer because there's no good screening for it. Good questions. So my question for you is, I'm gonna bring this up at the end. If any of you can think of how to adjust for clinical ascertainment bias, like I said, we can measure clinical ascertainment bias. We can measure about how much the clinical ascertainment bias is by doing this. This can tell us about how much clinical ascertainment bias is. But I haven't figured, I'm not a good enough statistician to know how to be able to say, well, if this is the clinical ascertainment bias, can we take clinical data and get any meaningful risk estimates from that data? I mean, yes, of course, we can get meaningful risk estimates, but can we get better risk estimates that adjust for that clinical ascertainment bias? Because most of the data we're using now is that there's fewer and fewer studies that are being funded to do prospective follow-up with people. And one of the reasons why there's fewer and fewer studies is there's more and more studies, more and more funding going towards, well, we just have big data. And let's just have this big data and do retrospective analysis with big data. Not thinking about kind of big data from all these hospitals might not be might have these biases. So if you can think of this, please come find me, and I'd love to put you on a grant. I don't have any funding now, but I'd love to put you on a grant application to figure it out. Thanks so much.
Thank you.